Hello BIO178. So in this video I will be explaining the different options that we have available for you to choose for your group projects. So there will be 7 options that I'm going to be describing here, and ultimately what we'll do is choose 5 of those. So I'm giving more options than we'll actually be working with, but at the end of the day we will be working with 5 of these different projects throughout the rest of the semester based on what you all are choosing. So the first option is doing metabarcoding microbiome analysis of 2 different related species of intertidal snail, Tegula galena and C. funebralis. So this is brand new data that I just got relatively recently as part of the workshop I participated in this summer related to DNA subway. I sent in some samples from my lab, and they did high throughput metabarcoding sequencing to gain some insight into the microbiome of these 2 different species. And so in these descriptions, I'll be giving you some information about the project itself and also about the analysis that will need to be done to get results from the data. So basically what we're doing is building on those skills we've learned in lab so far this semester, and then using those programs or software packages to analyze new data. So this particular project, you would be using that purple line of DNA subway that we've used previously, and you would be looking at the microbiome differences of these 2 different species of snail. I'll explain a little bit later why this comparison is relevant, but basically these are 2 different species that are found in the intertidal zone in Southern California, but their numbers have been adjusting. So T. galena is this spotted or stripy one here, C. funebralis is the purple one, and I've been seeing just anecdotally some differences where it seems like T. galena is increasing in the intertidal, but what we don't know is anything about the microbiome. So that's one potential option. Another potential option is doing the same sort of metabarcoding microbiome analysis, but comparing 2 different things. So in this case, this potential project option is comparing individuals of the same species of C. funebralis, but this time, instead of comparing it to a different species, it's comparing different populations from Northern and Southern California. So we've worked with this data set before where we were looking at data from 6 different populations along the California coast. For this particular data set, I have samples from 1 northern population, Slide Ranch, and 1 southern population, Bird Rock. And you would be using the same kind of data analysis, DNA subway, we've done before. So I think I've talked about this a little bit. One of my master's students is currently working on a project to see what the role of the microbiome is in thermal tolerance in the red abalone. So here with the different geographic populations of C. funebralis, I've talked about this a lot with my own research. I've looked at it in terms of genome-wide SNPs, I've looked at differences in gene expression, but I don't know at all if the microbiome of these 2 populations is different. So that's one option for a project as well. Another option, and I have to acknowledge that this is not my own data, I don't work with black bears, but this data and the following project were generously provided to us by Jillian, who is a previous master's student from Sac State, and she currently works at the forensics lab for California Department of Fish and Wildlife. And another person who helped us get this data is Joy Gaines, who actually was a student in this BIO178 class the first time I taught it several years ago, and now she's also currently employed at Department of Fish and Wildlife and doing a master's in my lab actually. So this project is looking at sex bias dispersal of California black bears. So what we have is microsatellite data from 5 different populations that are listed here. And for this analysis, you would be doing an analysis in RStudio where we used that higher FStat package when we were comparing the dispersal of male and female shrews. That's the graph I'm showing here. You would be doing the same type of analysis with this new microsatellite data and looking at different California populations of black bears. Another similar option is doing the same type of sex bias dispersal analysis, but this time in a different species, the California mule deer. So this would again be with microsatellites, and you would be comparing different populations, slightly different populations in the bear, and you would again be using RStudio to conduct the same type of sex bias dispersal analyses using that higher FStat package. That's another option. And then back to the snails. So you do have some options that are not related to my research, but most of them are because that's the data that I have. So this is again related to those different tegulus species, T. galena and C. funebralis. And the reason why I'm focusing on comparison of these two species is because I found that at several different sites in Southern California, the abundance of this C. funebralis population has really declined. So one of the first years I was at Tech State, I took some of my research students, we drove all the way down to La Jolla near San Diego, because that's the collecting site I'd been using for years during my PhD. And of course we got down there and there actually weren't any of these guys anywhere, which was great. Made me look like an awesome advisor. We went all the way down there and there were no snails. So there were no C. funebralis, but there were a lot of these guys. So this is that other species, T. galena, and their abundance seems to have increased at some of these Southern California sites. And so this particular project would be analyzing some heat stress survival data that we have gathered in my lab. So we've done some heat stress trials, we have some survival data, but we haven't looked at it across time like we did in that lab where we used the survival package. So you would be doing a similar type of survival curve analysis that we did with the red abalone survival data in lab previously. Another option, building off of this same type of data, same species, and other things we've done in lab, would be looking at the genetic diversity of individual C. funebralis populations. So in lab previously we've used RStudio and that diversity package to compare the diversity, a variety of different diversity metrics between Northern and Southern California. But I have actually never calculated and compared the genetic diversity of individual C. funebralis populations. So I've always been grouping all of these northern populations together, but it's and all of the southern populations together as well, but it's very possible that they are doing different things as separate populations. I've never actually examined that. And that's especially relevant if we think about what I've said previously about comparing to other species, especially if we're seeing some what are called local extinctions, where it looks like the abundances at some of these individual sites are really decreasing. So what's interesting is that this La Jolla site is where I said we went several years ago and couldn't find any of the C. funebralis individuals. This Bird Rock site is only like 7 kilometers away, and there are tons of them there. So there's something interesting going on. So looking at genetic diversity of these individual populations could be informative for future work. And then the last option is using the program PopArt that we used in lab. Remember, we built that haplotype network using that data from Dr. Ballerini with the different sequences from the plants that she collected. So in this particular project option, you would again be working with data from C. funebralis. But what you would be doing is constructing a haplotype network based on some sequences from a mitochondrial gene, NADH4, that I have. And we don't know exactly how or what the role is, but it seems like this mitochondrial gene has some kind of function in heat tolerance. We found some differences between Northern and Southern California, and there were some other signatures of selection on other mitochondrial genes, so we don't know exactly how or what pathway it's involved in, but it seems like this gene has some kind of role in thermal tolerance. And so what we could do is construct a haplotype network, and then use that to help us figure out for these populations here in Southern California. So remember I said that we're seeing big reductions here in this La Jolla population. The last time I went to this Aliso Beach site in Laguna Beach, there weren't really any individuals there either. And so if we're wanting to think about, like we talked about in the conservation genetics module, would it make sense for us to move individuals from a different population into these sites where we're seeing these large decreases in abundance? So I have data from these 3 populations and also from another site called Abalone Cove that's near LA. And so I have sequences for individuals from all of these different sites. You could build a haplotype network and then see what the distribution of haplotypes is and see if it makes most sense for individuals from one geographic region to be moved in to facilitate reintroduction for this species. So those are the project options. If you have any questions about what the data would look like or what the data analysis would entail, please reach out and let me know. Otherwise, you can go ahead to the next portion of this module, which will have instructions for you about how to actually sign up and choose which of these project options you would like to work with.